Today we are going to discuss about meiosis. It is a type of cell division in sexually reproducing organisms that reduces the number of chromosomes in gametes, the sex cells or egg and sperm. During meiosis, each diploid cell undergoes two rounds of division to yield four haploid daughter cells, the gametes. There are two phases, meiosis I and meiosis II. When meiosis starts, each cell contains two copies of each chromosome, one from each parent. Here in this cell, you can see four chromosomes or two pairs of chromosomes. Each chromosome consists of only one chromatid. During the S phase of the cell cycle, there is DNA replication. That means it creates a copy of itself. The chromosome before DNA replication has one chromatid, and after DNA replication has two chromatids. The two chromatids are firmly attached to the centromeric region. During the leptotene stage, those duplicated chromosomes, each consisting of two sister chromatids, are condensed from diffuse chromatin into more visible chromosomes. In the zygotene stage, homologous chromosomes, one from mother and one from father, come together. A protein complex called the synaptonymal complex connects the homologous chromosomes. This tight pairing of the homologous chromosomes is called synapsis. During the Pachytene stage, one chromatid of each homologous chromosome overlaps or crosses over a corresponding non-sister chromatid of the other homologous chromosome at one or more points. These crossing points are called chiasma. In the diplotin stage, synaptonymal complex dissolves and homologous chromosomes separate from each other except at the sites of crossing over called chiasma. In diakinesis stage, the nuclear membrane of the cell starts to disappear, and the appearance of fully compact homologous chromosomes is seen. The homologous chromosomes in a bivalent are still connected by at least one chiasma. Tetrad or bivalent consists of two homologous chromosomes, each with two chromatids. Important event during pro-metaphase is the development of a protein complex called a kinetochore around the centromere. Spindle fibers extend from poles on either end of the cell and attach to the kinetochores.
So during metaphase, homolog pairs, not individual chromosomes, line up at the metaphase plate for separation. This may seem familiar from mitosis, but there is a difference. Each chromosome attaches to spindle fibers from just one pole of the spindle, and two homologs of a pair bind to spindle fibers from the opposite pole. In anaphase, the spindle fibers contract and pull the homologous pairs, each with two chromatids, away from each other and towards each pole of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart. I hope that now you have understood how the chromosome number reduces to half, as we have started with 4 chromosomes in a cell, but now the daughter cells contain only 2 chromosomes each. In telophase, the spindle fibers break down, the nuclear membrane reforms, and the chromosomes return to an uncondensed state. Cytokinesis, the cell divides and forms two daughter cells. Both the daughter cells move from meiosis I to meiosis II, but there is no DNA replication. The cells that enter meiosis II are the ones made in meiosis I. These cells are haploid, have just one chromosome from each homolog pair, but their chromosomes still consist of two sister chromatids. During prophase 2, the chromosomes begin to recondense and spindle fibers begin to form once again. These spindle fibers attach to the sister chromatids. The centrosomes move apart and the spindle fibers forms between them. In this stage, the spindle fibers attach to the kinetochores of sister chromatids. The sister chromatids are arranged at the midpoint of the cells in metaphase 2. The two sister chromatids of each chromosome are captured by spindle fibers from opposite poles. In anaphase 2, the sister chromatids are separated. In telophase, spindle fibers disappear. Nuclear membranes form around each set of chromosomes and the chromosomes decondense. During cytokinesis, the cytoplasm splits in two and the cell divides. Meiosis begins with one parent cell. After the first division, there are two daughter cells, and then those each split, resulting in a total of four daughter cells. Four haploid cells in which each chromosome has just one chromatid. The gametes produced in meiosis are all haploid, but they are not genetically identical. As it turns out, there are many more potential gamete types than just the four shown in the diagram, even for a cell with only four chromosomes. The two main reasons we can get many genetically different gametes are crossing over and random orientation of homolog pairs. Thank you for watching and subscribe to the channel for more educational videos on biology.